This is the module for Biochemistry of Human Milk. My name is Kimberly Prezioso, and I'm a registered dietitian nutritionist and a certified lactation educator. Since you've already heard my bio, we're going to go ahead and get started. I wanted to just start with this quote. The unique feature of human milk is that virtually every component examined plays some extra nutritional role. The elegance of the system is remarkable, the more so we learn about it. Human milk is always changing, and yet it is always perfect. I want to emphasize that this module really only provides a broad overview of the biochemistry of human milk. In some ways, your text goes into greater details, and in other ways, it doesn't cover quite as much as this lecture does. What I do want to emphasize again is that if you do have any questions, you can always email me at kprezioso@gmail.com, and I'd be happy to talk to you. The composition of human milk is the biologic norm for infant nutrition. Human milk contains many thousands of distinct bioactive molecules that protect against infection and inflammation and contribute to immune maturation, organ development, and healthy microbial colonization. Its varied bioactive factors include cells, anti-infectious and anti-inflammatory agents, growth factors, and prebiotics. Unlike infant formula, which is standardized within a very narrow range of composition, human milk composition is dynamic and varies within a feeding, diurnally, over lactation, and between mothers and populations. Influences on compositional differences of human milk include maternal and environmental factors and the expression and management of milk, for example, its storage and pasteurization. Understanding human milk composition provides an important tool for management of infant feeding, particularly of fragile, high-risk infants, as well as for understanding the potential impact of storage and pasteurization on milk components. Further, we note some bioactive components found in human milk that are being developed and tested for potential medical applications as prophylactic or therapeutic agents. The study of biochemistry of human milk also provides a strong argument against the use of formula. It also plays a strong role in the present and future health of the mother. By the end of the session, the student will be able to characterize changes in milk composition that occur in response to infant growth requirements, explain how milk is a dynamic substance that changes in composition to meet the unique needs of the infant, describe key nutritional and bioactive components of human milk, describe the difference between the milk of a mother whose baby is born premature compared to the milk of a mother whose baby is born term, and compare and contrast commercial infant formula to human milk. In this module, we will be covering components of milk and their function, changes in milk composition, key nutritional and bioactive components of human milk, and comparison of human milk to manufactured milk. There are 3 stages of lactogenesis, which you will be covering or have already covered. What is important to know for this module is that milk synthesis is controlled by the degree of fullness and emptiness of the breast in lactogenesis 3. Empty breasts attempt to refill quickly at a rate of about 58 mL or around 2 ounces an hour. Full breasts secrete milk slowly at about 11 mL or 0.33 ounces an hour. Babies consume an average of 67% available milk per feed. Therefore, the degree of fullness and emptiness of the breast determines the rate of milk synthesis. As mentioned earlier, one incredible feature of human milk is its specificity. Human breast milk is a complex matrix with a general composition of 87.5% water, 3.8% fat, or 50% of total energy, 1% protein, and 7% lactose or carbohydrate source. The fat and lactose, respectively, provide about 50 and 40% of the total energy of the milk. Together, they contribute around 90% of the calories. The composition of human breast milk is dynamic, as we've mentioned earlier, and changes over time. It adapts itself to the changing needs of the changing child. For instance, during each nursing session, the milk that is expressed first, the foremilk, is thinner with a higher content of lactose, and this satisfies the baby's thirst. Following the foremilk, the end milk or the hind milk is creamier with a much higher content of fat for the baby's hunger needs. Variations are also present with the stage of nursing or age of infant. Variations are also present with the maternal diet, maternal health, as well as environmental exposure. And we'll discuss that in detail later. Most of these components vary over time. For example, the fat content of human breast milk can vary with maternal diet and is also positively related to weight gain during pregnancy. Remarkably, it's been observed that a mother's breast milk is almost always adequate in essential nutrients for her term infant's growth and development, even when her own nutrition is inadequate. And this is something that is important to remember when you're speaking with clients. This means you don't need a perfect diet to breastfeed. The food supply over history has never been, and in many parts of the world still is not, stable. This protects the infant against changing conditions and starvation. This does not mean that we shouldn't encourage and support women to do their best to improve the quality of their diet and that of their families. So going through the slide, you can see that we'll be discussing water, proteins, non-protein nitrogen compounds, carbohydrates, fats, fat-soluble vitamins, water-soluble vitamins, cells, minerals, dual-performing constituents, and anti-infectious agents, as well as additional variations. 87.5% of human milk is water. All other components, even the lipids, are dissolved, dispersed, or suspended in water. Even in hot, arid climates, human milk provides all the necessary water for a baby's needs. As we look at the nutrient composition in human milk, note that we will address different nutrients at different levels of depth. Some nutrients will be covered extensively, and some only mentioned briefly. For example, I won't be talking about selenium at length, but we will discuss iron and vitamin D extensively in terms of looking at their function, content, sources, etc. Human milk contains the smallest amount of protein in the animal kingdom, which is why humans have the slowest rate of growth in the animal kingdom. The milk composition of each animal species is directly related to the growth and development needs of each species. Remember that protein is comprised of chains of amino acids, long chains of hydrogen, carbon, oxygen, and nitrogen atoms. The nitrogen, and in a couple of cases sulfur, is what makes it different. Protein is used for growth, development, immunity, as well as for energy. The protein in breast milk provides all of the essential amino acids. Essential meaning those are nutrients that our bodies cannot produce. We have to obtain them from the diet. You'll hear the term essential amino acids as well as essential fatty acids. The protein offered in breast milk is effortlessly digested and easily absorbed into the bloodstream. The distribution of each specific protein is perfectly matched to the growth of the human infant, allowing the young child to use the protein with exceedingly high efficiency. Colostrum has nearly 3 times more protein than the quantity found in mature human milk, and regardless of the mother's nutritional status or dietary intake, the protein content remains stable. The protein content of milk obtained from mothers who deliver preterm is significantly higher than that of mothers who deliver at term. Protein levels decrease in human milk over the first 4 to 6 weeks or more of life, regardless of the timing of delivery. For feeding preterm infants, the lower level of total protein and specific amino acids from donor, typically term late lactation milk, alone is limiting and requires additional supplementation. Again, although human milk protein concentration is not affected by maternal diet, protein increases with maternal body weight for height and decreases in mothers producing higher amounts of milk. There have been several studies about the protein concentration of human milk, and on average it seems to be around 1.4 to 1.6 grams per 100 ml during early lactation. Then it decreases to 0.8 to 1.0 grams per 100 mLs after 3 to 4 months of lactation, and then it drops even further to 0.7 to 0.8 grams per 100 milliliters after around 6 months. However, again, other studies have found that protein is significantly higher and some lower. So again, it is variable. Formula manufacturers have tried to replicate human milk in order to mimic its performance. However, formula lacks certain proteins present in human milk, one of which is lactoferrin, which we will cover in detail later and which we've talked about a little bit in previous modules. The animal protein in formulas come from cow's milk, which is tougher, more rubbery, and more difficult to digest than the protein found in human milk. It is interesting to consider that the ongoing attempts by formula companies to revise their formulations highlights the very fact that formulas in their current state are less effective neurologically, nutritionally, or immunologically when compared with human milk. The proteins of human milk are divided into two factions, the whey faction and the casein faction, each of which is comprised of a remarkable array of specific proteins and peptides. The most abundant proteins are casein, alpha-lactalbumin, lactoferrin, secretory immunoglobulin A, lysozyme, and serum albumin. Non-protein nitrogen-containing compounds, including urea, uric acid, creatinine, creatine, amino acids, and nucleotides, comprise around 25% of human milk nitrogen. Whey is the clear portion of milk protein, and casein is the more solid curd component. If you remember the nursery rhyme, the line curds and whey, well, that's what we're talking about here. Whey is what gives a white, cloudy, or opaque property to milk. It forms soft, flocculent, or fluffy-like, easily digestible curds, and whey is the more easily digestible between the two. Artificial milk's protein, as mentioned earlier, are from cows, or they're bovine. Cow protein is much less digestible than the casein and whey from human milk, and casein is the dominant protein in cow's milk, or bovine milk. The quantity of casein is much lower in human milk as compared to other species. The whey to casein ratio varies with each stage of lactation, going from 90% whey to 10% casein in early lactation, 60 to 40 in mature milk, and 50-50 or half and half in late lactation. Artificial baby milks differ in whey to casein ratio. For example, Good Start from Nestle has 100% whey to 0 casein. Their rationale is that by removing all casein, the formula is easier to digest and more similar to human milk. Although human milk has casein, just it's human casein, not bovine casein. I mentioned earlier that human milk proteins are more digestible than cow milk proteins. Other formulas include Enfamil Lipo with iron from Mead Johnson, which has a 60 to 40% ratio. Abbott Nutrition, the Similac Advanced with iron, has a 48 to 52 ratio. Now, keep in mind that when you look at human milk, the whey to casein ratio varies over time. Again, much more predominant in whey in early lactation, moving to 50-50 in late lactation. The formulas that I just mentioned, for example, the 60-40 ratio or the 48 to 52 ratio, those don't change over time. That combination is present in early lactation, in mature milk, and in late lactation. It doesn't address the needs of the changing infant's gastrointestinal system. While some people may think that it's a good thing that Good Start has all whey, if you look at how the whey to casein ratio is changing over time, you can see that as a baby's GI system is getting more and more mature, the whey to casein ratio is in effect prompting or assisting the baby at learning how to digest more challenging foods as the baby's gastrointestinal system gets older. So it becomes more and more challenging gradually over time as the baby's gastrointestinal system learns to digest more and more difficult proteins. Other proteins found in the whey faction of breast milk are mucins, and mucins are milk fat globule membrane proteins. What those proteins do is they actually surround the lipid globules in milk, and since human milk has a very high concentration of water, that helps the lipid globules in milk be absorbed and digested a little more easily. There are a number of efforts to replicate the components of human milk in order to duplicate its immense physiological advantages. Overly assertive patenting of certain human milk components, followed by the genetic engineering and harvesting of them through questionable means, is commonplace in the attempt to pursue the unobtainable advantages of human milk. Let's look specifically at some of the whey proteins. Alpha-lactalbumin, which is the major whey protein, regulates milk synthesis. Its mucins bind pathogens and can kill cancer cells. It also provides calories and is a source of amino acids necessary for protein synthesis. Bovine milk is higher in beta-lactoglobulin, which is not found in human milk. Moving on to lactoferrin. Lactoferrin is an iron-binding protein found in the whey fraction. It hinders the growth of iron-dependent pathogens found in the GI, which protects against infections. Lactoferrin competes with the bacteria for the iron. So lactoferrin is involved in iron transport and absorption. It's also an antibacterial because it competes with bacteria to bind iron. It's an essential growth factor for B and T cell lymphocytes. It promotes growth of lactobacilli, the beneficial bacteria. It's produced in mammary epithelial cells, milk ducts, and other regions of the body, speculated to have local as well as systemic defensive properties. It also assists in regulating bone growth. When infants are given iron supplements, lactoferrin may be saturated, thereby rendering its protection weakened. Bacteria such as E. coli can proliferate and flourish. Offering any type of artificial or supplementary feeding can damage lactoferrin's protective effect. We talked earlier about what essential amino acids are, and essential amino acids mean that these need to be obtained from the diet. So there are 19 essential amino acids which are key for human development. 8 of them are present in human milk. Taurine helps develop the brain and retina. It acts in membrane stabilization. It's an inhibitory neurotransmitter, and it improves the fat absorption of preterm infants. It is not found in cow's milk. Tyrosine is actually very low in human breast milk, and phenylalanine is higher in bovine milk. More about the whey proteins. Lysozyme provides antimicrobial protection against different gram-positive bacteria by attacking the wall of the bacteria. Besides breast milk, lysozyme is found in saliva, tears, nasal mucus, and pancreatic juice. It is found in much higher concentration in human milk when compared to bovine milk. Secretory immunoglobulins, which are antibodies, are also a component of whey proteins. We've mentioned these earlier as well. So, secretory immunoglobulin A, which is the most critical immunoglobulin because it coats mucosal surfaces to prevent adherence and infiltration by pathogens, also neutralizes bacterial toxins. Maternal secretory immunoglobulin A also stimulates the production of infant secretory immunoglobulin A. There are other components, other immunoglobulins such as immunoglobulin G, M, and E present in breast milk. These secretory immunoglobulins are very high during the first days postpartum, and we have a slide later on which will actually show you how they transition over the first few days. But they're high during the first few days as the previously protected in utero baby is now newly exposed to pathogens. Studies have found that this may increase again during the second year of lactation when young toddlers are exploring the world and becoming more exposed to pathogens due to now, again, the extreme hand-to-mouth behavior. The antibodies delivered via maternal milk do not harm the beneficial bacteria that is being produced by the infant's own GI system. They only work against the harmful bacteria and can help prevent inflammation by competing with the other immunoglobulins for antigens. Looking at enzymes, there are more than 400 enzymes that have been discovered present in the whey fraction of human milk. They have multiple functions, only a few of which include aiding in digestion, which is especially helpful during the transition period when the baby is adjusting from placental to oral feedings. They also are compensatory digestive enzymes, which means they can help offset a negative effect. They also stimulate neonatal development. Some of these enzymes are lipases, which digest fat. They lead to superior fat and mineral absorption. And there are a number listed in your text, and we won't go into those here, but just know that there are a number of different lipases. And again, the LIP stands for fat, and the ACE stands for enzyme. So when you see anything with an ACE on it, you know that that's considered to be an enzyme. There are also amylases for carbohydrate digestion. There are alkaline phosphatase, and peroxidases, which act like hydrogen peroxide and destroy bacteria. And these are just a few of the different enzymes that are in human breast milk. There are also hormones in the whey protein faction of breast milk. There's prolactin in milk, which is different than the prolactin derived from maternal serum prolactin. There are prostaglandins, which have anti-inflammatory properties. There's oxytocin, adrenal and ovarian steroids. There are relaxin and insulin, and there are also thyroid hormones. There are also growth factors in the whey faction of human breast milk. There's epidermal growth factor that aid the GI and other tissue development. There's nerve growth factor, which may help central nervous system from birth-related injury. And there's also insulin-like growth factor. The non-protein nitrogen compounds, urea, creatine, creatinine, uric acid, glucosamine, and alpha-amino-nitrogen, as well as nucleic acids, nucleotides, and polyamines, have metabolic and immune functions, and they contribute about 20% of nitrogen. So we're coming up to this module's first test your knowledge slide. A mother asks you if her 3-month-old needs supplemental water. What would you tell her? Breast milk satisfies all water requirements. If babies are thirsty, breastfeed. The foremilk of breast milk will supply their water needs. Over time, what happens to the whey to casein ratio in breast milk? Does it increase, decrease, or remain the same? It decreases over time. The whey to casein ratio varies with stage of lactation from 90 to 10 in early lactation, which is super easy to digest, 60 to 40 in mature milk, which is still easy to digest, and 50 to 50 in late lactation, making the GI work a little bit. Carbohydrates are the primary energy-supplying nutrients consisting of chains of carbon, hydrogen, and oxygen. The principal carbohydrate of human milk is the sugar or simple carbohydrate lactose. The concentration of lactose in human milk is the least variable of the macronutrients, but higher concentrations of lactose are found in the milk of mothers producing higher quantities of milk. Interestingly, lactose is unique to mammals. It does not show up anywhere in nature. 40 to 45 to sometimes even 50% of the baby's needs are satisfied from lactose, and 7% of human milk is lactose as compared to 6.5% lactose in horses and kangaroos and 4 to 5% in cows, goats, and sheep. And again, this just emphasizes the sweetness of human breast milk. The concentration of lactose increases by 10% over the first 6 months. Lactose is a disaccharide, meaning 2 monosaccharide sugars are linked together, and those 2 monosaccharides are galactose and glucose, and those are synthesized in the breast. The synthesis of lactose begins at lactogenesis 2, about 30 to 40 hours postpartum. The increase in lactose secreted in the cell draws water by osmosis and affects milk volume. Of all mammals, the human milk is highest in lactose. Our brain is actually the largest in size, and our milk is the sweetest. And again, about 7% of lactose is found in human milk, about 7.2 grams per 100 milliliters. Lactose is integral in promoting newborn growth. It also plays a role in enhancing calcium as well as iron absorption and supplies the brain's sole fuel, glucose. It also provides galactose, and you'll remember glucose and galactose are the 2 monosaccharides that are linked together to form lactose, the disaccharide. And galactose is used in galactolipids for strong brain development, supporting the development of the central nervous system, as well as inhibiting the growth of harmful intestinal pathogens. Because it is digested slowly, lactose provides a steady release of glucose into the bloodstream. Sucrose, the sugar more commonly used in artificial milks that substitute soy for milk protein, is sweeter, but it splits more rapidly. This results in a high peak of glucose in the infant's bloodstream. They've also found that dental caries are higher in infants who are given formula due to sucrose. The absence of lactose in terms of health, growth, and developmental consequences is not clear. Just to emphasize, it's not just the presence of sucrose in formulas, but it's also the absence of lactose, which is not beneficial to growth. Just to note that primary lactose intolerance is extremely rare, and a high degree of lactose tolerance is generally exhibited by infants of all races. Lactase, and again, you remember that the ASE or the ACE at the end of a word indicates that it's an enzyme. So lactase is the enzyme that it's produced prolifically from the ages of around 2.5 to 7 years of age, and it's a brush border intestinal enzyme present by about 24 weeks gestation. You can see the picture on the slide. That's an example of brush border villi. Those finger-like projections are what absorbs the nutrients in our gastrointestinal system. Lactase is present along the brush border. At 2 to 11 months postpartum, concentration is 2 to 4 times higher than at the newborn stage, and lactase quantity is genetically determined. There are other carbohydrates in human milk, and the other significant carbohydrates are the oligosaccharides or polysaccharides, and those comprise about 1 gram per deciliter in human milk. Again, that depends on the stage of lactation and maternal genetic factors. The oligosaccharides are among the non-nutritive bioactive factors that I will cover when discussing immunity. Non-nutritive means they don't supply calories. They comprise the third largest solid component in breast milk. They are also some of the most diverse. More than 200 different types have been found. The oligosaccharides stimulate Lactobacillus bifidus and other beneficial bacteria by being calorically available. They also prevent bacteria from adhering to GI surfaces and protect against enterotoxins in the gut by binding to bacteria. Oligosaccharides possess anti-infective properties against pathogens in the infant GI tract, such as Salmonella, Listeria, and Campylobacter, and they do that by flooding the infant GI tract with decoys. These decoys bind the pathogens and keep them off of the intestinal wall. Oligosaccharides also have prebiotic capability by having a vital role in the development of a diverse and balanced microbiota, and this is essential for appropriate innate and adaptive immune responses and help colonize up to 90% of the infant biome or healthy GI system. We discussed bifidus factor earlier. Some of the polysaccharides are a component of bifidus. For example, oligosaccharides plus glycopeptides equal bifidus factor. With lactose, lactose bifidus, which occupies the intestine as a beneficial bacteria, is promoted. It grows and subsequently crowds out harmful pathogens. It also produces an acid that inhibits the growth of pathogens. Studies are being conducted to look more closely at the role of fructose in infant growth and development. And as we mentioned earlier, that as a component of lactose, galactose is also present. Fat is the most highly variable macronutrient of milk. The composition of human breast milk, as we've said, is dynamic and changes over time, and it adapts itself to the changing needs of the growing child. Variations are also present with the stage of nursing or age of infant, the maternal diet, maternal health, and pregnancy fat stores. And pregnancy fat stores are actually more easily accessed than other stores. Environmental exposure also plays a role. For example, severe malnourishment may be an issue, but in the vast majority of women, this is not a factor. Fat can vary widely from feeding to feeding, from mother to mother, and it even varies in one 24-hour period. For example, the calories in human milk can fluctuate by as much as 29%. Lipids change during the length of a feeding. Hind milk, which we covered earlier, which is defined as the last milk of a feed, may contain 2 to 3 times the concentration of milk fat found in foremilk, which is the initial milk of a feed. Foremilk is thinner and bluish with a higher content of lactose and contains around 1.5 to 2% fat, and this foremilk is what satisfies a baby's thirst. And again, remember that supplemental water is unnecessary during the first 6 months of life. This is milk that is accumulated in the breast since the last feed, and when an infant sucks and oxytocin is released, that stimulation allows the foremilk residue to wash down the walls of the ducts and ductules. This supports the importance of allowing babies to breastfeed on demand and end the feeding at will. Mothers should not restrict the amount of time baby spends feeding because important fats, protein, and calories are available, even more so at the end of a feeding. Hind milk is creamy and white with a much higher content of fat for the baby's needs, as well as to aid in sleep. It has about 2 to 3 times the fat content at 5 to 6% fat. Almost 1/6 of a baby's energy can be obtained during minutes 11 to 16 of a feeding. And that's, again, why it's critical for mothers to be encouraged to let infants remain at the breast and not limit their time. Protein levels tend to fall throughout the feed. The fat content also changes throughout the day. Research has demonstrated that milk fat content is significantly lower in night and morning feedings compared to afternoon or evening feedings. Other studies have found that around 25% of the variation in lipid concentration between mother's milk may be explained by maternal protein intake. The fat content of a mother's diet also comes into play, especially in regards to long-chain polyunsaturated fatty acid. Long-chain polyunsaturated fatty acids are transferred from mother to baby in the 3rd trimester through the placenta and to infants through breast milk after birth. During the last trimester and neonatal period, brain tissue is rapidly synthesized. Cell differentiation and development of active synapses in the brain need specific requirements of specific essential fatty acids. And you'll remember that essential fatty acids mean that we have to obtain them for food. We cannot make them efficiently on our own. One study has showed that the fat content and the percentage of all polyunsaturated fatty acids in breast milk increased significantly between the 6th week and 6th month of lactation. There is a lot of research being conducted right now on the role of essential fatty acids in human breast milk, and especially in the area of maternal dietary influences, as well as in preterm birth. We've already discussed that in the case of a premature birth, the transmission of these fatty acids is interrupted from the placenta to the fetus during, again, that critical last trimester. Studies have showed that decreased postnatal essential fatty acid blood levels in preterm infants are associated with neonatal morbidities or death. Therefore, after birth, the preterm infant is dependent on an adequate diet for sufficient fatty acid levels and may need supplementation. Visual acuity, visual attention, and cognitive development are improved in the addition of essential fatty acids to preterm infant formulas when compared to infants not receiving supplementation. When breastfed and formula-fed infants are compared, there's enhanced development of visual function, improved retinal function, and improved visual acuity in breastfed infants due to the long-chain polyunsaturated fatty acids. That is one reason why formula companies are aggressively pursuing the inclusion of these in formula. Other studies demonstrate an improvement in visual attention, problem solving, and global development. Even more studies exhibit the increase in developmental scores at a year of age when compared with infants that are breastfed 6 months or longer. That is attributed to either fat-contributing energy, brain composition, or both. Research has examined the association between maternal characteristics and the composition of human milk macronutrients and found that after 4 months postpartum, the macronutrient concentrations of human milk are associated with one or more of the following factors: maternal body weight for height, protein intake, parity, return of menstruation, and nursing frequency. This study also found that mothers who produce higher quantities of milk tend to have lower milk concentrations of fat and protein, but higher concentrations of lactose. We've also discussed the effect that breast fullness as well as emptiness has to fat content. As mentioned earlier, fat levels rise significantly in the 2nd year of lactation, adding yet another evolutionary layer to human milk. Even the age of the mother may play a role in the fat content of colostrum. Women who are 35 years of age or older may contribute more fat. Now, this may be due to greater fat synthesis and transfer to maternal milk, lower water content, or some combination of both. In any case, this may be welcoming information to older mothers. I'm putting the word older in, verbal quotes, as generally it becomes less and less easy to lose weight as years pass. So what is the composition of fat? The fat content of mature milk is 41.1 grams per liter with a range of 22.3 to 61.6 grams per liter. This is independent of breastfeeding frequency and is directly related to relative fullness or emptiness of the breast. Again, as a breast empties during an individual feed and or over the day, the proportion of fat increases. 70% of the fat variation in human milk can be attributed to this factor. An empty breast may have a greater fat content. A fuller breast may have less. The ratio of saturated to unsaturated fatty acids are fairly stable at 42% saturated and 57% unsaturated. The levels of some essential fatty acids in milk are decreased with tobacco use. Lipases, again, these are enzymes which aid in fat digestion, break down long-chain fatty acids. Free fatty acids are pathogenic. They defend against bacteria, parasites, and can inactivate viruses. 98% of lipids are enclosed in globules, ensuring less clumping and greater efficiency. Triglycerides predominate in human milk over short-chain fatty acids, long-chain fatty acids, and cholesterol. Lipases break down triglycerides into free fatty acids. There are also phospholipids and sterols included in the fat fraction of milk. So the additional functions of fat are flavor and satiety to breast milk, growth regulation, inflammatory responses, immune function, and they have a role in the development of motor systems. Now, numerous studies provide support that early exposure to human milk may be linked to lower blood cholesterol concentrations in adult life. And cholesterol has distinctive metabolic effects. It plays an essential role in all membranes and is a vital component of brain tissue, aiding the development of the myelin sheath, which is involved in nerve conduction. Breastfed babies typically have a higher cholesterol level than formula-fed infants. What is the primary carbohydrate in breast milk, and what are 3 of its benefits? Lactose is the primary carbohydrate in breast milk. The 6 benefits that I've included mean that lactose contributes to humans having the largest brain size. Human milk is the sweetest of all mammal milks. Lactose enhances calcium as well as iron absorption. Lactose helps supply the brain's sole fuel, which is glucose. Lactose inhibits the growth of harmful intestinal pathogens, and lactose is also digested slowly. Name 3 ways that fat content varies in breast milk. Fat content varies in the length of feeding, foremilk versus hind milk. There are daily changes to fat content, morning versus afternoon versus evening versus night fat content changes. There are fat content changes in terms of the dietary intake of the mom. The length of breastfeeding over days, weeks, months, and years, the fat content changes. The fat content changes when a breast is full versus when it's emptied. Fat content varies according to the length of time between feedings as well as the age of the mother. This is the end of Part 1, Biochemistry of Human Milk.